The bone-like cartilage, also covered by connective tissue from outside and inside. The connective tissue covers the outer surface of the bones. This part called periosteum. It's function like a precondylium. And also from the inside we have endosteum. Periosteum is usually thicker than endosteum and rich in the blood vessel. And you can see the collagen fiber from the periosteum penetrate into the bone matrix and attach firmly the periosteum to the bone matrix. These collagen fibers, usually collagen type 1, called perforating fibers or Sharpie fibers. Let's magnify the periosteum. Here you can see the periosteum has two layers, inner and outer layer. Outer layer is rich in collagen fibers, so we can also say the fibrous layer. But the inside layer is contain a lot of cells. It's rich in cell, or we can call it the cellular layers, especially osteoblasts, osteoprogenitor stem cells, and also the bone lining cells. All of these cells we have in this layer, and they are very important in bone repair. You can again see here the endosteum is a very thin layer of connective tissue. You can see covering the bone trabecula. All of these protrusion, or we can say extension of the bone into the marrow cavity called trabecule. This trabecule is covered by endosteum. And also you can see a lot of interconnective cavities between these trabecules. When we magnify endosteum, you can see endosteum, unlike the periosteum, is very thin layer. In fact, it's a monolayer. It's a one layer of the cells plus very thin and delicate and sparse matrix around the cells. Usually the cells are osteoblast, osteoprogenitor cells, osteoclast, and also bone lining cells. These cells compose one layer and cover the bone from the inside, line the bone from the inside. Usually the cell in this layer have a gap junction and adherent junction with each others. And we never call them epithelium because we don't have any basement membrane here. We just have a cell adhesion here without a basement membrane or basal lamina. So we cannot call them epithelial cell. You can see they are very similar to epithelial cell. And the bone is very hard because of the styloid matrix. So we cannot cut it into a very thin layer. We need to observation under the microscope. So for preparing a very thin layer of the bone, we have two methods. The first method is decalcifications. It means we have to first soften the bone by a decalcification solution, for example like EDTA, citric acid, and other acid, but with a low concentration we can use it for the calcification of the bone. By this solution, by this method, we remove the mineral part of matrix, so the remaining part is actually like other connective tissue, is flexible, pliable. We can cut it into the very thin layer, and after staining you can see the details of bone in the slice or in that section, the cells, the matrix. But usually By removing the mineral part, we just leave the collagen fibers type 1 in the matrix. This collagen, you know, the collagen gives the acidophilic property to the matrix. So usually we have the red matrix in the calcified slides. Another way is to prepare a very thin layer using a polishing stone. By the polishing stone, we polish the bone until the thin enough for viewing under the microscope. But this method called ground section. By polishing, we prepare a very thin layer of tissue without staining, without any fixation and other preparation, routine preparation. We prepare a very thin layer and see under the microscope a view like this. You can see the Haversian systems and cells and even canalicular and other component within the bone. Let's go to the classification of to classify the bone. The first is classifying the anatomy. You can see in the anatomy we can say we have a long bone, short bone, flat bone, and irregular bone. But and also the long bone, from the anatomical view, we can see have a three-part. A middle cylindrical part called diaphysis, and two bulbous end or outcrossing end called epiphysis. Two epiphyses, and the middle part, middle shaft called diaphysis. But again, by a gross observation in the cross section of the bone, we can see two kind of bones in the olive bones, long, small, and also the flat bones. In the outer, we have a thin layer of the compact bone. We see a heavy and dense bone in the outer part. Usually the compact bone forms about 80% of total bone mass. And in the middle of the bone, we have a spongy bone or cancellous bone or trabecular bone. Three names for this bone. And this bone also makes about 20% of total bone mass. You can see here with large magnification, compact bone, or sometimes called cortical bone, because we have on the surface in the peripheral part, and cancellous or spongy bone with a lot of interconnecting cavities between these trabecula. Again, here you can see the spongy bone. All of these cavity filled by a bone marrow, and all of these trabecular covered, lined by endosteum. Again, here you can see two compact bone filled between two compact bone by a spongy bone. Just in the flat bone, the spongy bone between the two compact bone or two plate, we call the diploe. Just in the flat bone, the spongy bone called diploe. But under the microscope, we have two types of bone: lamellar bone, woven bone, or lamellar bone or mature bone or secondary bone, and woven bone called immature or primary bone. The lamellar bone contain a regular arranged component, but woven bone contain irregular arranged component. Let's see in detail. But first, let's go to the carpenter. You can see two woody board here. The preparation of this board is very easy. You need to just cut the tree, for example, and prepare the wood like this, the board like this. Usually in the previously the carpenter prepared a door, window by this kind of board. But today we have another kind of board called plywood. You can see this board have several layers. It means the carpenters prepare this kind of board by attaching, by pasting several thin board together and make a thick board like this called plywood. Why the carpenter usually today using this kind of board instead of this board? This board is very resistant to fracture, to cracking, and also they are very resistant to water. Why? If seen in detail here, you can see each seam board in this plywood have a special directions or spatial orientation of fibers. The red and black arrow here indicate the orientation of fiber in each board, and when we pass this board together, you can see the fiber orientations change orthogonally in each successive board, and after putting them together, at the end we have a plywood like this, so it's very resistant to fractures. I think the carpenter and maybe the scientists learn how to make this kind of board from our bones. In the lamellar bones also we have a multi-layer, multi-lamella, multi-parallel sheet. So let's see these layers in the compact bone from the long bone. We have 4 types of these lamella in the compact bone. The first one you can see we have immediately under the periosteum and around the marrow cavity, some of them around the blood vessel, make a Haversian systems, and the other lamella, especially irregular lamella, filled between the Haversian systems. In this slide you can see the name of these 4 types of lamella. The outer parts, usually we have several layers of the bone called outer circumferential lamella. Around the marrow cavity you can see inner circumferential lamella. Usually we have a very thin or a few number of these layers around the marrow cavity. Around the blood vessel or Haversian canal we have a Haversian lamella, and filled between the Haversian canals or Haversian systems by a irregular lamella called interstitial lamella. So 4 types: outer circumferential, inner circumferential, Haversian, and interstitial lamella. Let's see in this schematic picture from the bone. You can see the periosteum and 2 layers of the periosteum, and perforating or sharpie fibers. Outer circumferential, or here called external circumferential lamella. Inner Haversian lamella, and filled between the Haversian we have an interstitial. You can see this one. Actually the interstitial lamella is remaining of the old Haversian lamella that digested by osteoclast. It's remaining part of that or previous Haversian systems. When we bring out the layer of Haversian system, for example here 3 layers, we can see in each layer we have a different orientation of collagen fibers. For example, here go from the left and down to the up and right, and in the next one you can see it's vice versa. The orientation in each layer is about 90 degree. It's about a 90 degree, or we have orthogonally orientation in each or in successive layers. And this orientation give or increase the strength of the lamellar bone, like a plywood. And osteoblast, it means osteoblast in the bone produce a matrix in a circular manner, and the cells finally trap between the layers and produce osteocyte. You can see osteocyte between the layer. When we have a longitudinal section from the Haversian system, you can see we have a canal in the middle of Haversian system called central canal or Haversian canal. Within the Haversian canal we have a the artery, we have a vein, and also one nerve in each canal. And in addition to longitudinal central canal, you can see we have some transverse canal. This transverse canal connecting the Haversian canal together, and also to the marrow cavity from the other side to the blood vessel within the periosteum. This canal, this transverse canal, called perforating canals. Here you can see, in addition to compact bone, this picture shows the compact bone is a lamellar bone. When we have a section from the spongy bone, you can see also here we have a multi-layer. We have a lamellar bone and also the spongy bone, but without any Haversian system, without perforating of blood vessel into the lamellar bone, because we have a very thin layer of the bone in the trabecular. We don't need to perforate the blood vessel into the bone. For example, the cell here, you can get the nutrient from the bone marrow, because you know the bone marrow is actually a kind of filled by blood. So they can take nutrient from the blood here and pass it to the outer cells, and we don't need to perforating of blood vessel into the spongy bone. Outer boundary of each Haversian canal called cement line. In this line we contain high amount of non-collagen protein in addition to collagen and also the minerals. Here we have many non-collagen protein here, and usually we see on the slide as a dark line around each Haversian system. I will show you in some slides. Again, here another picture show the circumferential lamella, outer, inner Haversian, and interstitial between them, and you can see the osteocyte trap between the layers. Central canal or Haversian canal, Volkmann or transverse or perforating canal. The name of transverse canal. Again, here you can see the layer of the Haversian system or osteon. Another name for Haversian system is osteon. Osteon means a unit of the bone. And you can see the orientation of collagen fibers changing orthogonally from the one layer to the another layer. You can see a real slide from the bone, Haversian system, Haversian canal, and I said around or outer boundary of each Haversian system we have a dark line called cement line. Again, here you can see the cement line around this Haversian system, the lacuna osteocyte. You can see singly occupied by one osteocyte. Because of a highly ordered or organization of the collagen fiber in each lamella, when we put the bone under the lamellar bone, special lamellar bone, under the polarized microscope, we can see two kind of layers, bright and dark layers, or bright and dark lamella. And the microscope show us the lamellar property of this bone. This double refraction or birefringence is because of the orientation of collagen fiber in each layer. Again, here you can see the lamellar bone under the polarized microscope. You can see a Haversian canal, Volkmann canal. Here you can see Volkmann canal connecting to Haversian canal here, and the black dot here shows us osteocyte within the lacuna. In the ground section from the bone also we can see the Haversian system, osteocyte, Haversian canal, Haversian lamella, interstitial lamella. We said it's the remaining from the digested, partially digested Haversian system by osteoclast. Magnify one part of from this slide, you can see Haversian canal, osteocyte, and also you can see, and clearly we can see the canalicules connecting two osteocytes together. This very good picture from the one Haversian system, osteocyte, and a lot of canalicule filled by a cell process, osteocyte process. In addition to lamellar bone, we said we have a primary bone. Actually, the primary bone is the first bone produced during the development and also during the bone repairment. It's the first bone the osteoblast produced at the site of, for example, in the fracture. Then this bone converted, removed by osteoclast and converted or produced new bone in the site of primary bone, or converted to the secondary bone or lamellar bone by osteoblast. So the primary bone or woven bone is a temporary bone, and this bone have the component of this bone arranged regularly, and you can see the interwoven area of the collagen fiber in this tissue. I compared these two bones in this slide. You can see we have many cells, or we have more cell in primary bone compared to the secondary bone. We have a few or a little amount of minerals in the primary bone compared to the secondary bone. You can see we have a high content of mineral in the secondary bones. And also you can see interwoven, irregular arranged collagen fiber in the primary bone, and a regular arrange of collagen fiber in a lamellar bone. Because of these regular arranging produce a sheet or lamella within the bone. But in some part of our body we have a primary bone, and these places, the bone in this place is never converted to the lamellar bone. The first place is between the flat bone of skull, head bones, called the suture, the kind of joint between the head bones. In this part, we always have primary bones filled in this area, and also in the site of attachment of muscle by a tendon to the bone. I mean, in this part, the site of attachment here we have a primary bone or woven bone.